I just want to start off by saying that it really is an honor to speak after Dr. Cervantes. However, I will make the point that even though the topic that I'm going to be speaking about is somewhat different, the essence of it remains the same. This is a talk about change. It's a talk about innovation. And I think above all, it's a talk about people and patients. So even though things are different in different areas of life, I think they also remain the same. I'm delighted and very grateful to the Center for Dialysis Innovation for inviting me to give this presentation. These are my disclosures. But I want to really start off with a recognition and a call to action. The recognition is for my very good friend, colleague, advisor, mentor, and role model for over 25 years, Jonathan Himmelfarb. I counted this up, Jonathan. We've actually known each other for 25 years. The call of action, though, is to all of us in this room, all of us outside of this room who are interested in vascular access. The first arteriovenous fistula was created in 1966 and reported in the New England Journal of Medicine. The group of patients that it was created in was completely different and much younger than the patients that we currently create arteriovenous fistulae in. About 12 years later, there was a report which clearly documented the first swing site stenosis in my mind that you can see over here. Archives of Internal Medicine, 1978. And yet, despite this, almost 50 years later, if you look at the HFM data, you find that if you place 100 fistulae, only about 40 will actually mature to support dialysis without assistance, and a full 34% will be abandoned. And I am going to make the case that this lack of true change, so the word change again, has resulted in poor value in vascular access care, in that we spend a huge amount of money and achieve outcomes that I would say are dismal, 50 to 60% arteriovenous patency at a year. And remember, this is without any mention of either quality of life or of equity in access to care. And I just want to expand upon that last point about equity, or rather inequity. We know that Black and Hispanic patients have lower incidence rates of fistula payment, and that this occurs regardless of insurance, which brings out a whole host of other potential reasons. We also know that Black patients do worse at every step in the AV fistula care continuum, from placement to maturation to unassisted fistula patency. So the big question, I think, before us as a community is, how do we create change in vascular access? And I think many of us, again, in this room and outside, have worked hard on trying to develop a patient-centered ecosystem for discovery and for process of care innovation in vascular access that goes all the way from biology to technology to patient care, from initial ideation and investment to the identification of druggable and deviceable targets, to clinical trial infrastructure and surrogate endpoints, and finally to regulation and reimbursement. And what's even more important is that we have to surround this innovation ecosystem, I would say, with equal measures of both equity and justice and access to care. So very similar again to what Dr. Cervantes spoke about earlier. What I'm going to do over the next 10 minutes or so is speak about some of these pillars of change for vascular access as shown on this slide. And I'm going to start off with biology. So this slide shows you the classical radiological presentation of dialysis vascular access dysfunction. In the left panel, you see a tight perianastomotic stenosis in an AV fistula. This is the lesion that often results in fistula non-maturation. In the right panel, you see a very tight stenosis of the graft vein anastomosis, which often results in thrombosis. Now, if you take tissue from either of these regions and look at it under a microscope, you get what we've called the classical histological presentation of vascular access dysfunction, which really comprises an aggressive neointimal hyperplasia, where you have a migration of smooth muscle cells, myofibroblasts and fibroblasts coming in from the media, and perhaps also the adventitia in response to both hemodynamic stress and surgical injury. I want to make two additional very important points. This whole process is likely exaggerated by the inflammation, oxidative stress, and endothelial dysfunction that characterizes uremia. And point number two is that the final amount of luminal stenosis is always as a result of a combination of neointimal hyperplasia together with inward or negative remodeling, or at least a lack of positive outward remodeling. Now, it's difficult to summarize everything about the vascular biology of dialysis vascular access in one slide. So I've clearly distilled it very, very much. But what this slide results in is, it results in a construct which says that vascular access stenosis is likely to occur as a result of two interactive upstream pathogenetic pathways, hemodynamics and uremic vascular biology, which then results in two downstream, again interactive, pathological pathways, the inward remodeling and the neointimal hyperplasia. And the reason that this construct is so important is that if we want to develop novel therapies for vascular access, it is essential that they target one or more of these pathways in my mind. But even more important than the biology, and we're going back again, I think, to Dr. Cervantes' talk, is that these therapies need to be patient-centered. And the reason that they need to be patient-centered is that we know that the things that are important to patients are often very different from the things that are important to physicians or industry partners or federal agencies. We have a lot of data on this in dialysis, less so in vascular access, but we do know from this work by Charmaine Locke that physical complications in patients with a fistula, such as pain and cannulation perhaps, and disruption to routine were far more important to these patients as compared to infection, which most physicians would say is actually the number one problem. Having said that, though, I really do believe that this is an exciting time for vascular access because there are a large number of therapies in the clinic or in clinical trials that are trying to target biological pathways in a patient-centered manner. And these therapies go all the way from creation, percutaneous fistulae, maturation, the laminate scaffold perhaps, or the venous stent wrap, and finally maintenance through the use of drug-eluting balloons. Now, I know that Dr. Vasi is going to address many of these new innovations in the next talk, so this slide is just a mention of those. But what is even more exciting for me, and I think many others, is what is coming down the pike. And I am going to show you on this slide some of the innovations that we're working on in our own laboratory that go all the way from a smart magnesium-based biodegradable stent whose degradation can be turned on and off, to an endothelial protectant in combination with Alyssa Panich at Georgia Tech, to bioengineered vessels in combination or working with Yadong Wang at Cornell, and then very exciting in my mind, nitric oxide releasing catheter locks working together with Mark Schoenfisch at UNC. The great excitement when you talk about some of these novel technologies in the context of change in vascular access, though, is that they could allow us to achieve the K-DOKI's vascular access goals of getting the right access in the right patient at the right time. So what do I mean by this? Well, let's suppose in the future we had a 50-year-old with average veins, moderate comorbidities, well, this person needs an AV fistula, but AV fistula have this very high maturation failure rate. So in the future, this person could perhaps get a fistula together with a venous stent or a vascular device or some other innovation. And then if you have an 80-year-old with no veins, multiple and severe comorbidities, this patient, as appropriately described in the KDOQI guidelines, would probably do best with a catheter. But catheters have these terrible problems with infection and thrombosis. So in the future, the hope would be that this 80-year-old would be able to receive a catheter with an anti-infective and anti-thrombotic coating. But I would argue that this is just the beginning, because just as people are different, so too are their vascular access stenoses. And the vision that we would have for the future is a vision where we would be able to stratify both our patients and their stenotic lesions using direct RNA sampling or advanced imaging such as IVUS and optical coherence tomography, such that in the future, we would be able to say that lesion A in this patient needs to be treated with drug-eluting balloon Z, while lesion B needs to be treated with drug-eluting stent Y. And what would be even more important is if we were able to say that lesion C has such a bad prognosis that you really need to create a new access, while lesion D is probably going to respond really well to plain old balloon angioplasty. And the importance of this is that it allows us to risk stratify in vascular access care such that we would be using novel therapies in the patients who would benefit the most from them, and thereby we would be reducing both resource utilization and cost. But what about the vascular access of the future? So there's no question at all that technology can change clinical paradigms. So if tomorrow the Center for Dialysis Innovation comes up with a port that never gets infected, never gets thrombosed, never causes central stenosis, and allows patients to take a shower, then that would become the vascular access of choice. But as we think about the vascular access of the future, you can't really give a talk in Seattle without asking the question about what's going to be the right access for future dialysis modalities, for portable, wearable, and implantable dialysis. And the technology roadmap on innovative renal replacement therapy by the Kidney Health Initiative did actually look at it, and I've pulled out a line from it, which says that we need to develop vascular access with internal connection to the native vasculature that maintains patency at low flow rates without the need for systemic anticoagulation. And we're clearly not there yet. I've spoken a lot about discovery innovation, but many of the problems in vascular access are clearly due to process of care issues. So there's a huge role for process of care innovation. So let's suppose we decided on a group of people who we wanted to get a fistula to be working before they started dialysis. The process of care isn't rocket science. We need early referral to the nephrologist. We need vein preservation programs. We need to follow up the fistula and refer for angioplasty and surgery as needed. We need in the future, obviously, to use new therapies. And then very, very importantly, when the patient is ready for dialysis, because this entire process needs to be done during the CKD stages, then it's essential that we use an expert cannulator to do the first cannulations. Unfortunately, as all of you are aware of, there are huge barriers at each of these steps, but I would argue that each of these barriers is actually a huge opportunity for local process of care innovation. I also want to emphasize that it's not just about innovation and patient-centered technology. You can have the best idea, the best biology, the best device, but if your pathway for drug development is not clear, if you do not have the right regulatory endpoints, if your reimbursement pathway is not clear, then this wonderful idea and therapy will never ever get to the patients who are desperately waiting for this innovation. And that's why projects such as a previous Kidney Health Initiative project on clinical trial endpoints for vascular access trials is so important. And this project actually came up with 2 papers on endpoints for arteriovenous dialysis access and for dialysis catheters. And there was also an FDA regulatory perspective. And again, I would recommend this to all of you in the audience who are interested in the area of vascular access innovation. Our hope, of course, for the output, such as the KHI papers, is that they would serve as a catalyst to enhance innovation in this field. We hope that these papers will make it more attractive to industry and academia to come into this field, for people outside the kidney space to come into this area. And we hope that it will generate investment in this area. So the 3 I's, if you will: interest, investment, and innovation. And then finally, about policy, so important. The Advancing American Kidney Health Executive Order has placed an emphasis on global payment systems and on value-based care. This could include giving a lump sum of money to care for a fixed number of patients with CKD4 and end-stage kidney disease, and potentially share in either the profit or the loss. We strongly believe that global payment plans may actually be able to incentivize novel therapies in vascular access. So let's suppose you had a device that enhances fistula maturation. It costs $3,000, but saves $30,000 in downstream costs by preventing angioplasty and surgical requirements. Well, in an eat what you kill fee-for-service system, this may not be that attractive, but this device becomes extremely attractive in a global payment system because the savings would obviously result in overall profit. So how do we put it all together? So I really do believe that we have a good handle on the pillars of change for vascular access. Despite this, vascular access continues to be a huge clinical problem. And I would argue that this is because our implementation of what we know is poor. Vascular access care remains fragmented to a very great extent because no one truly owns it. And very few people or groups have approached this problem in a truly holistic manner. And so what the ASN wants to do in a new initiative that it has just started called Transforming Dialysis Access Together is to create a true platform or a meeting place for the entire dialysis access community. Patients, nurses, technicians, coordinators, physicians and surgeons of every stripe, LDO leaders, innovators, payers, regulators, funders, researchers, investors. And then once we get all of these people in different groups, but occasionally all together, then we want these people to address dialysis access care in a holistic, cross-cutting manner, going all the way from education and awareness to policy and payment, to equity and advocacy, to innovation and entrepreneurship, and obviously to also address issues such as data generation and guidelines. We've outlined some initial priorities and strategies that are shown on this slide. Obviously, we want to improve patient care. We want to develop the next generation of dialysis access. We want to cultivate the ideal workforce. And we have strategies for this shown on the right. But what I really want to emphasize is that this is a bottom-up big tent initiative. We want to get people together. We want to have ideas bubble up from the dialysis access community, from the kidney community, from the medical community. We want to serve as a convener. We want to get people from outside of medicine into this area. And if we can do that, that's when we're going to be able to change, using the word change again, dialysis access together. So finally, this is the Ideas Conference, and I thought I would end with really a children's storybook, which is titled, What Do You Do with an Idea? And my goal in showing this video is really to enthuse and to reach out to all the innovators and entrepreneurs in this audience. Because in innovation, as in life, you really have to believe. And here we go.
What do you do with an idea? One day I had an idea. Where did it come from? Why is it here? I wondered, what do you do with an idea? At first, I didn't think much of it. It seemed kind of strange and fragile. I didn't know what to do with it, so I just walked away from it. I acted like it didn't belong to me. But it followed me. I worried what others would think. What would people say about my idea? I kept it to myself. I hid it away and didn't talk about it. I tried to act like everything was the same as it was before my idea had showed up. But there was something magical about my idea. I had to admit, I felt better and happier when it was around. It wanted food, it wanted to play. Actually, it wanted a lot of attention. It grew bigger and we became friends. I showed it to other people, even though I was afraid of what they would say. I was afraid that if people saw it, they would laugh at it. I was afraid they would think it was silly, and many of them did. They said it was no good. They said it was too weird. They said it was a waste of time and that it would never become anything. And at first, I believed them. I actually thought about giving up on my idea. I almost listened to them. But then I realized, what do they really know? This is my idea, I thought. No one knows it like I do, and it's okay if it's different and weird and maybe a little crazy. I decided to protect it, to care for it. I fed it good food, I worked with it, I played with it, but most of all, I gave it my attention. My idea grew, and so did my love for it. I built it a new house, one with an open roof where it could look up at the stars, a place where it could be safe to dream. I liked being with my idea. It made me feel more alive, like I could do anything. It encouraged me to think big, and then to think bigger. It shared its secrets with me. It showed me how to walk on my hands, because, it said, it is good to have the ability to see things differently. I couldn't imagine my life without it. Then, one day, something amazing happened. My idea changed right before my very eyes. It spread its wings, took flight, and burst into the sky. I don't know how to describe it, but it went from being here to being everywhere. It wasn't just a part of me anymore. It was now a part of everything. And then, I realized what you do with an idea. You change the world.
So with that, I again would like to thank all of you for your attention, and I really look forward to the discussion that I hope will follow. Thank you again.